is the last part of Unit C, protein structure and function, and this is under advanced. So if you were still struggling with the beginning of this unit, you would want to leave this alone to maybe later on in the year, maybe at the midterm or the final, you'd be ready for this. So key ideas of this screencast are going to be looking at two key ideas in Biology 12. First one is that the function of a protein is determined by its shape. And that lovely key that we're seeing right there, and we've seen in every screencast, is the same exact idea, right? It has a structure, and the structure of this key is what gives it its functionality. It can open a door. It can start my car. Our chair, our lazy boy chair that we want to sit in comfy wumpy watching our TV show. The structure, the 3D structure of this chair provides its functionality. That's why we buy it. That's why we're willing to pay a lot of money for a comfortable chair. The structure is really well designed for its function. And a protein, because it has a specific shape, is going to have functionality due to this shape. A protein shape then is going to be determined by its primary structure, so by the order of the amino acids. And we want to start to understand and think about the DNA's code, the genetic code that resides in the nucleus in our genes, coded for by DNA, is what codes for this primary structure. So some big ideas. So some vocabulary, 3D shape. So 3D shape just means its conformation, its overall structure when it is finally built and ready to go to work. Protein structure, we want to recall, if we've forgotten, that there's 4 levels: primary, secondary, tertiary, and quaternary. Quaternary being an optional level. Not all proteins need to have these to function. So primary is going to be linear, secondary, the alpha helix or the pleated, beta pleated, and tertiary is the final 3D shape. Denaturation, when a protein structure is destroyed, and sickle cell anemia is the example that we will look at in this podcast. Let's go. So again, because it's so important, I want to be sure, if you didn't catch it, understand it, about protein structure. There are 4 levels going from simple to complex. And we want to make sure we understand that these structures are consecutive, meaning when I see a protein being built, when the amino acids are being assembled on the ribosome, this is the primary structure. And DNA's code is what provides the information on the primary structure of a protein. The primary structure is what will ultimately determine the tertiary, the final 3D shape of the protein. And this ultimately then is what determines how this protein will function in our body. So the primary will appear linear. Peptide bonding between the amino acids is what maintains the primary structure. The secondary is when there is interaction between the backbone of the amino acid chain due to H bonding, and it turns into an alpha helix. 3D shape is as a result of some interactions between the R groups, and the protein begins to fold over to become globular, a big glob. And this is like our brown lazy boy recliner, the final 3D shape. Quaternary, more than one polypeptide is bonded to another. And hemoglobin is an example of a protein with quaternary structure. So some examples, well, proteins, we say they're so important. What are some examples? So there are numerous examples. The one that we will concentrate on in our next unit is looking at enzymes. However, there's other examples that just to understand how important proteins are, we want to look at some. So there's some structural proteins in our body. In our skin, we have collagen. Nowadays, women are willing to pay money and go and get collagen injections around their lips to give them more plumpness and to retain the look of being youthful. As we age, unfortunately, these proteins start to stretch and sag due to gravity, and the elasticity from the elastin isn't so great, just like in a sock, a tube sock that loses its elasticity over time starts to sag. So does our skin. Keratin, a structural protein we find in hair and nails. Another example we've already talked about, insulin is a hormone. It is a protein. So hormone describes its function. It is a protein, and remember, insulin was an example of a protein we talked about in the secretory pathway. Another example of a protein functioning, really important, is the Y-shaped protein made by our immune system in response to a foreign particle entering our body, an antibody, which we'll talk about when we talk about our blood. Transport proteins, and the one that we're looking at as an example, is hemoglobin. Hemoglobin is a large protein. It occupies our red blood cells and it functions to transport oxygen. So pretty important to our life, right? Oxygen is how we make ATP in our mitochondria. With glucose, we burn them and produce ATP energy. So how do we get the oxygen? The hemoglobin. We rely on the hemoglobin to bind to oxygen and transport it through our blood. Other examples, enzymes like salivary amylase made by our salivary glands is found in our saliva, the watery substance that we release into our mouth. It is a hydrolytic enzyme breaking down starch into maltose, and all enzymes are proteins. So an example of how key structure and function in a protein is the example of sickle cell anemia. And sickle cell anemia, unfortunately, is a genetic disease that affects the hemoglobin that transports oxygen in your red blood cells. So due to a mutation in DNA, so DNA's code has a mutation in it, it causes there to be a switch out of one amino acid. So these unlucky folks have a mutation that results in valine replacing glutamic acid in hemoglobin. So we're seeing this would be a normal, healthy person, and this would be the person with the mutation for sickle cell anemia. What happens is this causes the red blood cell to be misshapen, and this in turn affects how the red blood cell can move about. So it doesn't carry, the misshapen red blood cell doesn't carry as much oxygen that gets stuck in blood vessels, and unfortunately sickle cell anemia people die at a young age from this disease. So this is a perfect example. One amino acid out of place causes this key protein to have the wrong structure and as a result not to function, with some pretty serious results. So denaturation is the word we use to refer to proteins when they have been their shape has been modified, and this is not due to genetic mutation. So sickle cell anemia was an example of a genetic mutation. Denaturation is a general word to describe a protein that has its structure misshapen, and this is going to be change in conditions. When I change the temperature, I change the pH, salinity, environmental factors in the environment or inside of the organism change, and this changes the shape, which will change the function of the protein. So hopefully you can describe the 4 levels of protein structure. You could give some examples of functions, and you could explain how protein structure relates to protein function.